The question I was presented with was to discuss the structure, folding, and pharmaceutical importance of membrane proteins using the principles, theories, and concepts we discussed in class. Starting with membrane receptor proteins. Receptor proteins are membrane proteins that aid in the communication between the inside and outside of cells. Signaling molecules like hormones, neurotransmitters, and cytokines interact with the receptor and trigger some sort of internal response. Cell adhesion molecules are involved in anchoring cells to one another, typically through interaction with other adhesion molecules of the same kind or with the extracellular matrix. They are often involved with the immune system response, and the interaction between these molecules and immune cells can trigger white blood cells to migrate throughout the body. Enzymes may also be embedded in the membrane, as was shown by succinate dehydrogenase. The target of NSAIDs, a class of zinc fillers, is cyclooxygenase, which is embedded in the membrane of the smooth ER. Ion channels help to regulate the flow of ions in and out of the cell and allow concentrations to be managed. A well-studied example is CFTR, which is the protein whose mutations cause cystic fibrosis and whose functions will be described later in more detail. Similar to ion channels, transporter proteins control the movement of substances into and out of the cells. However, they can use both active and passive transport, meaning it can transport molecules either with or against the gradients. Additionally, transporter proteins are for larger biomolecules, such as hormones and neurotransmitters. I have decided to focus mainly on ion channels and transporter proteins. In order to highlight the differences between these two, an active transport protein known as serotonin transporter, or SERT, will be discussed in detail. Membrane proteins are made like any other protein. They are transcribed, translated, and then folded. The difference is that their final destination is in the membrane. The main mutation that causes cystic fibrosis is a 3-nucleotide deletion that results in the absence of a phenylalanine. This leads to misfolding of the CFTR protein, which never reaches the membrane. The phenylalanine plays an important role in the interaction with the membrane and also with the N-terminus of the protein. This leads to insufficient chlorine ion transport, which is used to regulate the water content of mucous membranes, leading to thick and sticky mucus. In this case, there is no chloride ion transfer of any kind, but there is still sodium transfer, which is also used to regulate the water content, so it's not a death sentence. There are other mutations that may lead to cystic fibrosis. A missense mutation, meaning that one amino acid is replaced with a different amino acid, is also fairly common. This generally leads to reduced regulation of chloride ion concentration, which in turn leads to less severe cases of cystic fibrosis. One of the missense mutations is the replacement of a glycine with an aspartate at the 551 location. One can imagine the replacement of a large amino acid for the smallest amino acid would lead to a conformational change of the protein, leading to decreased function. It would make sense that membrane proteins make up for approximately 60% of pharmaceutical targets, due to their accessibility. One of the best-selling classes of drugs are the NSAID class, which target a membrane-bound enzyme in the smooth ER called cyclooxygenase. This enzyme catalyzes an early step in the production of a prostaglandin responsible for the inflammatory response. These drugs, NSAIDs, block the active site of cyclooxygenase, preventing arachidonic acid from fitting in. In Figure 1 of the supporting document, a drug can be seen in the active site, and the heme groups are also highlighted. Cyclooxygenase is actually part of a larger enzyme complex called prostaglandin synthase. There is a 2-enzyme subunit which has a peroxidase to activate the heme groups on one side and a cyclooxygenase on the other. 2 of these subunits make up the complex. SSRIs are another class of drugs which target a membrane protein. In this place, serotonin transporter, or SERT, is the target. This protein is responsible for transporting serotonin against the gradient by exchanging it with sodium and potassium. Normally, serotonin is released into the synapse, where it travels from one cell to another's receptor and then is released by the receptor and bounces back and forth before it is eventually transported back into the cell against the gradient and is removed from the cycle. SSRIs clog the transport chain, making it so that the serotonin cannot be removed from the cycle and can continue to activate the receptors. In Figure 2, Prozac can be seen clogging the channel of an SERT. That concludes the assigned question. Next is a question of my own making, namely, what does the cytoplasm taste like? First, the composition of the cytoplasm. The exact ion concentrations vary a lot between cells. Because of this variance, I'm going to replicate a neuron cytoplasm. so I think that cell looks the neatest. There are a few staples. Cytoplasm is mainly water. Reactions in the body occur under aqueous conditions, so the cytoplasm is 70% water. I will be using solid water. Potassium, which makes up the largest ion concentration of the cytosol, is in fact one of the most common elements in the body. In neurons, it is used to transmit electrochemical impulses in order to relay messages and is used elsewhere in the body for electrolyte balances and helps to prevent muscle cramps. In our cytosol smoothie, potassium will be represented by bananas, which is a pure and unadulterated source of potassium. Next most common ion would be sodium and bicarbonate ions. Though this is about 1/15 as common as potassium, bicarbonate is, of course, the main buffer in our blood, which keeps our enzymes happy and not all squiggly. Bicarb is formed by dissolving CO2 in water, which is what most of everything is made up of. To represent these ions, baking soda is added. Amino acids are also present in fairly high quantities. Amino acids make up proteins, and proteins do stuff. In order to make this the purest protein, all lipids are removed from peanut butter, powdered peanut butter. Upset. Magnesium is present in fairly low concentration and is used to stabilize the phosphate group in ATP. Here we crush up a magnesium supplement. Since this is a neuron, neurotransmitters are also present in the cell. One of the most common neurotransmitters in vertebrate is glutamate. In order for this to pass in my body, a sodium atom is added to this molecule. Ramen. Finally, present in scarce amounts of the cell is calcium, which is used for the neuronal synaptic transmission, so omitting yogurt. There's green bits in it. I'm really bad at pouring. Why am I so bad at this? Okay. It's a lot thicker than I thought it would be. Gotta be honest.
How does it taste?
Tastes like something. I'm not quite putting my finger on it though. It reminds me of that peanut butter pasta that everyone likes apparently. Or it's basically just peanut butter and soy sauce mixed together. Which is basically, I guess, what this is. And a banana.
And blueberry yogurt.
Ow. Got my tongue. Okay.
not a fan.
It's basically like a peanut butter, banana, and blueberry smoothie with that someone like vomits into.
So are we done?
I think I'm done.